So thanks very much for the invitation to come and speak with you today. I changed my title slightly, so to really, what I've been thinking about over the last little while is sort of what we've been doing over the last 10 years in the field of high content screening and functional genomics. So it's not just what we've been doing, but the field itself of high content screening and functional genomics is about 10 years old. And so it's a good time, I think, to reflect and decide, did the field of high content screening deliver what it promised to do? Because we sure promised a lot 10 years ago. And now, knowing what we know, and now that we have good hardware, good software, and we can do these screens repeatedly and quite easily, and many labs can do them now, and versus a few labs that could only do it 10 years ago, should we keep doing them, and should we keep learning? Are we learning anything from them? So it's a time to kind of think about all these questions, or I could just be going through some sort of early midlife crisis or something, I don't know. Anyways. So we use high content screening to study signaling in cancer cells, and particularly we're interested in how cancer cells, for example, this is in a breast tumor, respond to different signals from their microenvironment. So if cancer cells, especially in tumor cells, don't exist alone, they exist in a community of different cell types. For example, there's stromal cells, up here in this region, as well as extracellular matrix components. And these have the effect of stiffening the tissue. For example, we know that tumors are palpably stiff. That's how a physician can feel them. A patient can itself feel them. And that's because the stroma and ECM is stiffening the tissue around it. And that stiffening is something that the cancer cells thrive on. There's also immune cells, which often infiltrate the tumor tissue. These cause inflammation as part of the immune reaction. And even though there's very much, immunotherapy is a very hot topic right now, it's also important to remember that immune cells and local inflammation is also something the cancer cells are feeding on. And so we're interested in how these signals from the tumor microenvironment, such as coming from the stroma and the extracellular matrix, particularly the mechanical signals from this region, as well as signals from immune cells, particularly inflammation, are being integrated by signaling networks in cancer cells to drive cell shape changes, which ultimately drive metastasis or even tumorigenesis itself. So we want to know essentially the architecture, dynamics, and dynamical properties of these different signaling networks and how they drive shape changes that cause disease in the first place. And it's important to remember, I think, that even when the cancer cells are gone, that these things like stiff tissue, damaged tissue, stromal tissue, and even immune cells can still be around that microenvironment, which can cause the patient discomfort even after you've successfully treated a tumor. So it's very important to understand these two different factors. Now, we are studying cell shape changes because it's very important to realize that if a cell is going to metastasize, it has to undergo multiple cell shape changes. It has to invade the local tissue, it has to get into the bloodstream, get out of the bloodstream, colonize a new tissue, and all of those are absolutely essential. You need to change the shape in order to do those things. They must change their shape in order to metastasize. We know, of course, that the frequent fact we hear that metastasis accounts for 90% of patient deaths. So our argument is that if we understand how cancer cells change their shape, this will open up new therapeutic avenues for the treatment and prevention of metastasis. Now, this is our specific problem, but we're using high-content screening to understand what I think is a very general problem, and that's really understand what's going on with signal transduction networks. And we can break the problems, the challenges in understanding signaling into three different components. The first is, which many people are interested in, and that's, I think, why functional genomics even started, is really what are the components? Who are the players in signal transduction networks? Just think of your own favorite signal transduction network, don't have to think about mine, that are relaying signals from the environment into a particular phenotype. So what are the components here of this pathway that say, okay, there's some signal here in the environment, and that's going to change the behavior of the cell. And we still, I think, frankly, don't know many of the components that in many signaling networks. We also want to know causality, and this is a big tricky one. So how do we know which component is regulating the actions of another? So we draw these arrows very freely, but how do we know that A is regulating B and that B is regulating C? And then the third challenge, which I think we need to think about more, which people don't really think about, is the fact that the architecture of these signaling networks can change. It's the dynamical properties of signaling networks. A signaling network might look like this in one state, but then switch over into another configuration, if you will, have different dynamics in another state. This could be in one shape, this could be another shape, this could be G1, this could be S, and we have very little idea of how this changes, one network changes in one state to another. And so I'm going to call this, I don't like this, but I'm going to call it rewiring, and trying to understand how networks change in different conditions. And this is all something we're trying to understand using high content screening and HCS-based approaches. So I think these ideas, these questions are common to many of us in the audience. So our workflow for doing this has really never changed. We have typically been using RNA interference, and despite the evolution of CRISPR, I'm still a big fan of RNA interference for a number of reasons. We image the cells using high throughput robotic microscopy, and then we use computer vision algorithms to segment individual cells. From each individual cell in our experiment, we generate what are called morphological signatures of up to 600 different features, describing each cell up to 600 different ways, trying to capture its shape. So what are those? Well, you can ask how big a cell is, for example. Where's the nucleus positioned with respect to the rest of the cell? We're absolutely obsessed with protrusions. So how many protrusions does each cell have? What's the angle between each protrusion? What direction are the protrusions facing? For each cell, we describe a number of texture features, which are peaks and valleys of image intensity, which give you a 3D feel, often from a 2D image. And we've developed a number of features which we call microenvironmental features, describing maybe not necessarily the shape of the cell, but its neighborhood. So if a cell is sitting in a colony, how many neighbors does it have? How much free space does each individual cell have around it? And each cell gets described 600 different ways. In a typical experiment, we might have hundreds to thousands of different RNAis. Each RNAi we might image hundreds to thousands of different cells. We might have, so over a huge database, we might have millions of different cells quantified 600 different ways over tens of thousands of different conditions. This generates large data sets, and I think where we do our best work is in this transition from the large data sets into network modeling. How do we take these large data sets and understand architecture, dynamics, network rewiring? And that's what I'm going to spend most of my time telling you about today. And once we've got these network models, we are very concerned about doing experimental validation, especially in vivo models. Here's our platform, not too much tech. So we use two Perkin-Elmer Opera QEHS high throughput microscopes. For fans of high throughput microscopy, these are the older generation now. I call them classic, not older. We have a Cell Explorer automated experimental prep platform, and it is, you invited me, so it's important to say that most of our RNA, all of our RNA dispensing is done now using the echo dispensers. And as you'll see, this does become an important part of our workflow, especially as the experiments get more and more complex, which they're going to do. So the first question we're trying to get, and this is again, one of the starting points of all sort of functional genomics, is trying to infer parts lists from these different screens. And we started to infer these parts lists using essentially ideas from conventional genetics. So imagine this is a shape, a wild-type cell, wild-type phenotype. Again, think about it as your own phenotype. It kind of doesn't matter. This is the wild-type form, and if you knock out a gene, for example, gene A, and it has this shape, it's big and flat, whereas this one is not big and flat. And if you might then be able to infer that A regulates the process, negatively regulates spreading. You get rid of A, the cells are able to spread. Now what genetics allows us to do is if you knock out gene B and you knock out gene C, and they have the same shape quantitatively using hundreds of different morphological features, you can say that A, B, and C are likely connected together in some complex, and that complex is likely regulating spreading. It's unlikely that the genes are working independently, and that's the power of genetics. Genetics allows us to do this. And we showed that this indeed can be true almost 10 years ago now by looking at local signaling networks. We call them regulating cell shape during migration. So knocking down a relatively small amount of genes at the time, 249 different genes, quantifying the shape of each individual cell in the data set, and then using different computational methods to reduce that data and cluster the different morphological signatures, we could describe about 35 different local networks regulating different aspects of cell shape. So for example, you knock down all the genes in cluster 6. Instead of those spiky cells I showed you, the cells become round. And it's very easy to intuit that you can just say, well, if they're round, it's probably that those genes are regulating protrusion, and in fact that most of them are. So that doesn't tell you anything about the genes you knew something about, but it tells you a lot about the genes you didn't know anything about. So now you can characterize all these new components of protrusion pathways. You can extend this logic further. If the protein products of these genes are localized at the protrusions themselves, at the edges of cells, we can predict where the unknown factors are going to be. They're also going to be at the edge, and in fact, we could show that. So we could show that if we didn't know where a protein was, we could predict very correctly that it would be at the edge of a cell and regulating protrusion. So just by looking at the shape of a cell, we could predict protein function, and we can also predict protein localization on a subcellular level. We have another interesting category of genes here. If we knock them out, the cells, again, have this big, flat, kind of scrambled egg morphology. And that's because the cells are spreading, but they can't pull themselves back off the matrix because they have a defect in adhesion disassembly. Now, what was interesting about this particular network is that mass spec studies had been done on the adhesion disassembly complex, and many proteins which make protein-protein interactions with each other are also enriched in this complex. So we can predict function, we can predict localization, and we can predict protein-protein interactions just by looking at the shape of single cells. And this allowed us to characterize hundreds of new genes which we really didn't know anything about before. And in this sort of beginning study, we broke each one of these complexes down into what we call local signaling networks that regulates different aspects of cell morphologies, for example, during the migration process. So a group of genes that would regulate, very active at the tip of cells, regulating the formation of initial protrusions, ones that would regulate adhesion, ones that would be important in tail retraction, for example. And this is really a powerful approach, which I have to admit, even we probably should use a little bit more because we really haven't yet to expand this to a whole genome-wide level. Now, one aspect about this study is that each one of these clusters represents a local network, or for the mathematically inclined here, a complete graph of what we call genes or proteins here. We don't know the directions in between them, who's acting upon whom based on this data. And there's been a lot of sleepless nights I've had over the last 10 years trying to think about how we can go from shape data to inferring causality between these different components. And most of those sleepless nights have resulted in absolutely no progress. It's been very, very difficult, I think not just for us, to get arrows between these different components and to break some of these connections here that we need to do. There's been some success in the field, but I would have to say it's been pretty limited. The best way we've been able to do it is by integrating other sources of information and by invoking this sort of integrative image omics, we call it now, to describe causal relationships. So if you start bringing in new pieces of information, then you can start breaking these graphs and inferring causality. So 2008, we showed that if you do high content screening, and especially if you do double RNAi screens and you integrate phosphoproteomic data derived from mass spec, you can infer relationships between kinases and substrates. Amongst a complete graph, if a kinase, I can predict the substrates of individual kinases in that cluster using these two sources of information. Along the same time, we just used, again, a double RNAi screen, some mathematical modeling, and basically common sense to say that to infer causality between GEFs and GAPs, which we know act upstream of GTPases. So in these two studies, we had to know something about some of the proteins. We had to know these were kinases, we had to know these were GEFs and GAPs, we had to know these were GTPases, and then we could start integrating information to get at causality using shape data. Our most successful attempt, I think, has been just very recently, where we've used high content screening data, phosphoproteomic interaction data, not our own, and RNAi transcriptional signatures generated by the Broad Institute. So knock down a gene, know its shape, knock down a gene, know its transcription, and that has allowed us to infer causality between many more different components. For example, not just GEFs, GAPs, kinases, but basically any protein, and especially do it upstream of transcription factors. So I think the theme here is that integration is really the only way so far that's successfully got at causality within these signaling networks. There is one other trick we've used to try to infer causality, and that was in a study where we were looking at transcription factors such as NF-kappa B, YAP, and the SMAD proteins. Now, if you don't know what they are. It doesn't really matter. They're just transcription factors. We know they cause the epithelial to mesenchymal transition, but they're just transcription factors doing something to cell shape, and we were trying to understand signaling networks upstream of these transcription factors. We used, again, high-content screening here as a starting point. So what we did was not just measure shape in individual breast cancer cells, but also measured the activity of different transcription factors based on their nuclear localization. So in breast cancer cells, if you don't have TNF-alpha, an inflammatory molecule, you measure the shape and you measure NF-kappa B. NF-kappa B will all be in the cytoplasm, and you can infer that it's inactive. You add TNF-alpha, all the NF-kappa B goes into the nucleus. You can infer that NF-kappa B is active, and you can also simultaneously measure the shape of the cell while you're also measuring NF-kappa B. So this was our starting point to get at this. Now, when we started looking at the data, we saw something which was initially quite unexpected. So if we looked at the shape of the cells, so these are a three principal component analysis of cell shape. Just think of it as shape space. Each one of these is a single breast cancer cell, and its position in shape space is the shape of it. So these are small round, these are big flat, for example. And then we've colored each single cell here by the amount of NF-kappa B in the nucleus, for example, after we add TNF-alpha. And what you can see immediately is that there's a striking correlation by the shape of the cell and the amount of NF-kappa B in the nucleus. We didn't necessarily expect this. We didn't expect there would be any relationship between NF-kappa B activity and cell shape per se. We could imagine it would just be all over the place. But seeing this correlation allows us to do something here. So we can use Bayesian methods to describe causality. So we've established that there's a correlation between shape and NF-kappa B. So for example, between a single feature, the roundness of the nucleus, we knew there was this correlation, let's call it 0.7, between NF-kappa B. Bayesian inference allows us to do one thing, and that's decide whether NF-kappa B is responsible for nuclear roundness. Is it the activity of this protein which causing the change in cell shape? Or is it the change in cell shape which causing the change in NF-kappa B? We predicted this, but it's not what we found at all. In fact, what we found is that cell shape was very much impacting the activity of NF-kappa B. So not just the nuclear roundness of cells, but the shape of the cells, the area of the cells affects the amount of NF-kappa B in the nucleus. Protrusions, roughness affects it. Cell roundness, eccentricity, center's distance. The amount of neighbors you have affects the amount of NF-kappa B in the nucleus in inflamed cells. So it's all these shape factors are impacting the amount of NF-kappa B activity. NF-kappa B is responding to cell shape, not the other way around. And we saw this across all breast cancer cell lines in all different conditions that we measured. So it was really that shape is really impacting the amount of signaling and not what we expected, not the other way around. So we built a model based on this that's interesting, and that the TNF inflammation, for example, in the tumor microenvironment, if it hits an epithelial type cell, NF-kappa B activity, and in fact the activity of other transcription factors is largely inhibited. But if it hits a more mesenchymal shaped cell, let's say a flatter moving cell, then this mesenchymal shape activates NF-kappa B. So cell shape regulates signaling and not the other way around. Now this has a very, very important implication for screening though, not just for our screens, but maybe for your screens as well if you're doing them. And that's that if cell shape is impacting your phenotype, you have to be very careful about how you're interpreting your phenotypes. So I'll skip that. So let's imagine we have a transcription factor here called YAP. We know that cell shape is affecting its activity, and let's imagine we're screening, chemical screen and RNA screen for the amount of YAP in the nucleus. Now, if we do that screen, and we do it across many different conditions, you might find some hits, for example, gene A, which lowers YAP activity. But it might be NF-kappa B, it might be SPAD, P53, it doesn't matter. You've got a hit based on your RNAi, following RNAi. But you've also got a shape change. Now, if shape is affecting signaling, this poses a problem. Because you might have a case where it's, as you might predict, that your gene is affecting your signal, such as YAP, which is affecting cell shape. Or you might have a phenomena where the A affects cell shape, which affects your output signal. So your phenotype, your hit, might be in fact due that you've changed the cell shape and not because you've changed your phenotype that you're actually measuring. So we had to think of, if this is true, we wanted to find ways to correct for defects in cell shape that could be induced by RNAi. And so we created a mathematical model, this is the mathematical model for YAP, that allows us to distinguish indirect from direct hits. So we screen, for example, for YAP activity in the nucleus. Again, it doesn't matter. Think of any phenotype you want. And we build a mathematical model that predicts what the phenotype should be based on the shape of single cells. Then we monitor the shape and the phenotype of interest across the entire screen. And then what we can do is find true hits, ones that are real that we should believe, or ones that have just changed your phenotype, but don't have any direct impact on what you care about, the pathway you care about. So these ones might change your phenotype. They might have been seen as direct hits, but they're not hits once you correct for their effects on cell shape. And so we showed this for YAP, but now we do it essentially for all our screens. Every screen we run now, we correct for the effect on cell shape, and hit lists just change really dramatically. And you can imagine why that might be. I mean, cell shape can affect many different parameters. The density can affect cell shape. All these things can really impact on the phenotype you're looking at. So I actually would even go further to say that this irreproducibility that has been seen in the RNAi screening field could be in part due to this failure to correct for these kinds of effects. So there's this common idea out there, which I'm not a huge fan of, but there's this idea that if you perform one screen, one lab performs one screen, one lab performs another, they perform another, the hit lists do not tend to overlap very well. And this has kind of been puzzling the field. People have accused it of off targets, et cetera. But it could be that some of the effect, I'm not saying all of it, some of the effect could be due to the fact that these different screens aren't correcting for cell shape. Different screens could have different cell shapes, different densities, different cell types, just slightly different cell types, which is affecting shape differently. And if you corrected for these kind of indirect effects, your overlap could improve. So that gave us that statistical way, again, a way to infer the flow, that just the flow was not what we thought it was. We saw that shape was in part of this flow and it was not upstream of shape. And so the last part of my talk, what I'm going to focus on now is how these networks can become rewired. And again, this is how we're getting these dynamic properties from very static data sets. So we were able to get into this idea, but with the observation that different phenotypes like cell shapes are heterogeneous. So this isn't a surprise to anyone that's looked at cells. If you just look at wild type cells, pick your favorite cell line, if you look at the shape, most of you might have already noticed that they have many of them will have different shapes. These are just wild type cells, and you can see, we just plotted them out. You see all sorts of different shapes. That's one observation, but the real more striking observation was this one, that if you over different RNAis, not just the average shape changes, but the amount of heterogeneity changes. So if you, for example, do a screen, and you see the shape change here in this RNAi A, they all have this shape, and it's quite homogeneous. But if you do you use RNAi B, they would have this average shape, but they're much more heterogeneous than A. That's easy to see. And if you look at C, the average might be here, but they are the most heterogeneous in this population. So gene knockdown is not just affecting your average phenotype, but it's also affecting the amount of heterogeneity present in the population. And that's different heterogeneities allowed us to do different statistical tricks to get at causality and also rewiring. So the first thing we needed to do when we were doing this is quantify the amount of heterogeneity present in these different cell types. So this is one cell type we were interested in, and we use over millions and millions of cells, thousands and thousands of replicates. We saw that essentially cell shape, or this particular phenotype, could be grouped into 5 different discrete forms. The bulk of the cells form this normal rounded shape, but there was also, these are just wild type cells, there was also this teardrop shape, a spindle shape, a big flat smooth shape, and a big flat ruffled edge shape. And no matter what you saw, we plate the cells out again tomorrow, I promise you, you'd see majority of this in this frequency, but you'd also see these subfrequencies. It wasn't this mass, it wasn't this mess, it wasn't stochastic, it wasn't random. The cells are basically programming themselves to have these 5 different subshapes. And if you look at RNAi, the subshapes change. So 30% of the time over knocking down all kinases that you see nothing, okay, fine. But then 40% of the time you see the same 5 shapes but in a different frequency. So now these 4 subshapes have started to increase. 30% of the time 2 shapes of the 5 dominate the field. Here, very rarely, about 6% of the time you see a shape in the middle of the 5, and only 1% of the time do you see a shape that's not considered part of the 5. So this was surprising because across this screen, we were essentially never creating new shapes. We were enriching for phenotypes that were already there to begin with. And again, think about it for your own phenotypes. Think about it for drug resistance or nuclear translocation. Well, all you might be doing for these RNAi screens or even chemical screens is enriching for phenotypes that were already there, present at a low frequency. Now, with this heterogeneity in mind and this gigantic data set in our hands, we could do something. We could start drawing maps in between these different shapes. Again, no dynamic data here. This is all fixed data. And we could draw probability maps of how one shape transitions to another. So each one of these arrows here describes the probability that from this shape, it will go to this shape. From this shape, it'll go to this shape. And so it's, again, not random. There's, again, a pre-programmed way that cells transition between these 5 shapes. And this is regulated. So the 5 shapes all exist. The paths are quite deterministic. And when you knock down genes, you can change them. So if we have wild-type cells, and this is the map, you knock down P10, now every other route becomes closed to these cells, and only this route becomes open when you knock down P10. So genes are controlling both the transitions and the number of shapes present in the population. Now, what does this have to do with cancer? Well, it seems, here's our idea. Well, we looked at different cell lines. For example, we know P10 was a particular, we knew very potent or tumor suppressor, I should say. If you look at P10 wild-type cells in culture, we were able to show that, in fact, we would see essentially one large shape, which corresponds to that dominant shape I showed you before. When you knock down P10, or P10 loss, I should say, we haven't knocked it down. These are just P10 deficient cell lines. Now, again, you see the spindle shape emerging. So back to this slide. Oh, I don't have that. So we're looking at the difference between the wild-type cells are basically all in the N form, but if we knock down P10, we're predicting that they would be in the N and the spindle form. And that is the case. So P10 null are basically in the percent of elongation describes the fact that you're seeing 50% enrichment of the spindle form. Now we knock down P10, you go from mostly Ns to mostly elongated and round. You put back P10 into cells that don't have P10, they go from a mix of 50-50 elongated and round to just round. And then we can do this in vivo in mouse tumors. If they have P10, they're mostly round. But if they've lost P10, as predicted, they're basically both round and elongated. And we find, this was all fixed data, I should say. But so now we can try to confirm this using a live cell assay. So these are melanoma cells sitting in a 3-dimensional gel. We're imaging them in 2D, basically, but they're, so it's 3D, 3.5D over time, because we're imaging them in 2D, but they're grown in 3D over time, which is 3.5D. And we were able to develop a segmentation algorithm which captured their shape live and follow these cells live in in this 3-dimensional matrix. And what we could see in P10 lost cells, the cells were essentially flipping back and forth between this round and elongated shape very quickly, in the order of minutes. And so our idea is that in the case of P10, you might have a cell that's non-metastatic, for example. There's some event where it becomes metastatic, and now they go engaged into slow cycles of protrusion and contraction. But if you have P10, you're going to be mostly sitting in this space, in this shape here. But if you lose P10, you now have this shape and this shape. And because P10 is such a commonly lost tumor suppressor, our idea is that this gives metastatic cells an advantage. It's kind of a bet hedging approach. Imagine if this form might be good, for example, soft tissues, might be good to invade soft tissues. It might be more drug resistant, and I'll show you more about that later. And this one might be better for stiff tissues. It might be better for division. But if you now split your population, you lose P10, and now cancer cells have a choice between these two different shapes. It might give them a survival advantage. And so now by increasing the frequency, you've got 2 different shapes to choose from instead of just one. And if a drug kills this one or it kills this one, you've always got that other one to rely on. This is bet hedging, this isn't. Now, this brought us to the idea that if we could start measuring these phenotypes in 3D over time, could we, of course, do a screen in 3D over time? We call it a 4-dimensional screen. It's still basically a 3.5D screen. And what we do is we take RNAi, we plate the cells on top of a collagen matrix, and then we screen. We can't do this in hundreds. We can do it in 100 or so different genes. Do the exact same thing, but now we're generating morphological signatures over time, doing network modeling and characterizing the role of genes. Now, I liked what we heard earlier about we have good assay technologies, but poor kind of prep technologies. These are, for lack of a better, I mean, these are a bitch to produce. I mean, this is 96 well, 384 well plates. We're putting in a very, very thick collagen matrix. We have to transfect RNAi into these cells, get the cells into the matrix somehow. The matrix tends to be very uneven. It tends to peel off. Prepping this is just been very, very difficult, and we still have yet to really do it. We can't do a very high throughput screen because a lot of this is done manually at this time using multi-channel pipettes. Now this part, as you know, we've heard, is a lot easier. We have technology to do this, and I'm going to show you even better technology we have to do this. But we were able to do it for at least a couple hundred RNAis, and what we could show is that we could generate not just statistical fate maps, but generate actual fate maps about how cells transition between these different shapes. So in this case, we found, again, now this is 6 different shapes instead of 5 in this particular cell line, and we could show that if a cell was in this shape, the star shape, the frequency of it hanging there was very high, but it had a high probability of going to this one, lower probability of going to this one, lower probability to go to this one. And if we take all the data, we can generate a map about how the cells are transitioning between the different shapes. Now, it's, again, not random. There's 2 routes. One is called the apolar route, and one is called the polar route. Now, you might be like, at this point, going, who cares about these different shapes, and who cares about these transition? But this has very clinical implications, because what we've just, sort of hypothesizing and sort of shown is that this state here is more of a dormant drug-resistant state, and this state here is more of a drug-sensitive state, and especially when I'm talking about BRAF inhibitors. So if a cell looks like this, and we have others have shown this, if BRAF inhibitors are placed on these type of cells, they tend to be more sensitive. These guys tend to be much more drug-resistant. Now, having these maps the idea would be, could we find ways to block these different transitions between them? If you block the cell here from going into this drug-resistant state, from making this switch, could make the population much more susceptible? And of course, we have RNAi data so that we can find these particular conditions which block these. So all these different genes we, for example, knocked down here, 20 different genes, multiple different RNAis. We can show that, for example, if you knock down this gene, RAC3, they tend to hang out in this more drug-sensitive, elongated state and don't move into this drug-resistant state. And the prediction would be these are going to be a lot more sensitive to RAF inhibitors than, for example, this one. And that's what we're testing now. Now, as I alluded to, we're trying to increase the throughput of these screens. So one way, and which I'd love to discuss today, is how we can do this prep a lot better, but still the imaging isn't wonderful. We're doing all these on operas, and I have to say it's been amazing that we've been able to get anything out of them. This is a hundreds of microns thick matrix, and we're coming from the bottom, so it's amazing we can see these cells on top of the matrix. So we've spent a lot of time recently trying to improve the imaging technology in order to get better cells at these. And with Chris Dunsby's lab at Imperial College London, we've developed a new high-throughput light sheet microscope for 3D imaging. So for people, physicists, optical physicists in the room, you can figure that out. I can't explain it, so don't ask me. But what this allows us to do, I think we've all heard a lot about light sheet imaging, but this is, to our knowledge, one of the first, if not the first, high-throughput light sheet microscope for 3D imaging. So again, we plate the matrix in 3-dimensional in wells, high 96 or 384, or even 1536 wells, put them here, put the cells on top, and now use light sheet microscopy to not just image a section of the well, which I was showing you before, but to image the entire well. And so these pictures are the entire well. I'm coming at it from the side here, and you can see, for example, the meniscus of the gel here and the cells sitting on top. So this allows us to project this coming at it, I call it from the side, and you can see different meniscus, and we could see, for example, if cells start invading into the gel. And we can also see them, of course, from the top. So this is the side view. You can see now we've put cells at the bottom of the plate, and we're now letting them invade into the gel. We can see cells very clearly invading into this gel sitting on top. And now looking at it from the top view, this is the entire well. We're getting high resolution imaging of the entire well. Just to show you that indeed it is high resolution, because these are huge files. It's not just like we're getting a scan. You can really see that if we just pull up a small part of these things and we're scanning through, we're projecting through the gel, we're getting extraordinary detail, light sheet level detail, I guess, of the different subcellular structures, but we're getting the entire well at the same time, and we can make 3D projections from that. So we're quite excited about this, but again, I get back to it, the prep is very difficult. So I'll end there, and just to summarize what we've talked about. So maybe, I don't know, I've decided that high throughput screening is a powerful tool that can be used to describe signaling network components, dynamics, and rewiring. And we're still doing it. We're still excited about it. So I think there's still a future for it, and I think pharma is thinking the same. But I think what we've learned over the 10 years is we can learn a lot about, that's what we learned over the 10 years, but we have to be very, very careful about many different factors. One of them is the shape of your RNAis or your compounds. What does it do to the shape of the cells, and what does that do to the protein pathway of interest? And we can use mathematical approaches to sort out indirect and direct effects. We want to embrace heterogeneity, so I think we're going to hear a lot about single-cell approaches in this meeting. We are very much into single cells. Everything we do is single cells, and we can use heterogeneity to, especially in combination with statistical analysis, to learn about signaling pathways. And I mean, I've been to a few HCS meetings already this year, and everyone is just obsessed with 3D and 4D. You hear spheroids are just, everybody's doing spheroids. I'm not sure everyone knows why, but clearly we're doing it. We do single cells, for example, but anyways, we're all moving into 3D, and I think 4D is the next step. We still need to improve the imaging hardware, the prep hardware, in order to get to these, address these challenges, and computationally, it'll also be a difficult challenge to deal with. So that's it. Just thank the members of my lab. Oh, very dark there. People that have done this. My funders, Cancer Research UK and Stand Up to Cancer, and the microscope work was done in close collaboration with Chris Dunsby, Hugh Sparks, and Vincent at Imperial College. That's it. Thank you.